Hello everyone, and welcome back to CBI Online's Simple Publications. Today we're talking about a paper on trypanosome motility, or in other words, parasites and how they swim. Recent research has shown some extraordinary and revolutionary concepts in this field, but before we get to those, let's start by looking at the parasites first. All throughout the day, we get attacked by all kinds of pathogens that cause illnesses, and these can be categorized in different groups: bacteria, viruses, fungi, and parasites. Parasites, as opposed to the other options, are just really tiny animals, sometimes single cells, that are small enough to move within our bloodstream. Some famous examples include malaria, tapeworms, and the ones studied in this paper, trypanosomes, which you probably know better from the creature spreading it, the tsetse fly. If you are bitten by a tsetse fly, you may be infected by the trypanosomes inside and develop sleeping sickness. Of course, this doesn't happen instantly. A bunch of stuff happens in your body before you get sick, not the least the defenses of your immune system kicking in. That's why it's crucial to know how the parasite behaves once it's inside the host. This allows us to learn how to better combat these infections. So what are trypanosomes really, and what do we know about them? Well, for starters, this is what they look like. All those tiny squiggles are trypanosomes. They're just tiny creatures. They can swim forwards and backwards, and you might think I put those arrows the wrong way around, but the side where the tail is, quote unquote, the flagellum, is actually the front. They have another type of movement called tumbling, where they just wriggle in place and don't really go anywhere. One of the reasons why studying movement is so important is that, by swimming, trypanosomes are able to fight off proteins from the immune system. Normally, your antibodies stick to an invading pathogen in order to communicate to other cells in your body, but, as you can see here, by moving, trypanosomes move the proteins to their mouth, where they can eat them before a further immune response is triggered. Now, it's important to understand that what you see here is blood that was just drawn from an infected carp and put in a petri dish, so it's easy to see what's going on. And that means that the bigger question remains, is this also what happens in the body? We know trypanosomes move through the body using the bloodstream, but they're also able to leave the bloodstream and move to other tissues. This is known as extravasation, and earlier research took this a step further by making tiny cylinders that would mimic red blood cells in the bloodstream to see how they would react to that, and, by using some known metrics about blood cells, they showed that the trypanosomes could bob and weave in between the blood cells in order to get where they needed to be. A second discovery that was made was that they can use the flagellum to stick to surfaces, which would likely be a first step in extravasation. However, scientists never sleep, and technology keeps pushing the boundaries. Every so often, this results in things being not quite as they seemed. With the development of new high-speed, high-resolution cameras, new kinds of studies became possible, revealing new information in the lives of these parasites. Let's have a look at what the researchers did in this paper. In this paper, they looked at an in vivo model, so you're looking at live zebrafish, and that means that, from the start, everything is different. Instead of looking at an artificially created environment, you're actually looking at what happens inside the animal. And there are a few really neat things that become apparent when you do this. For one, as you can see here, the environment doesn't quite resemble a flat plane with some blood cells in it for the trypanosomes to swim between. Instead, they are just swept along with the current or caught by the red blood cells zooming past. In the bloodstream, you just see them showing their tumbling behavior, but when you look at the tissues, you do see directional movement as, without all the obstacles and strong current, they're free to move around. A second important finding is this one. Here you see the trypanosome attached to the wall of the blood vessel, and it might be a bit difficult to tell, but if you look closely, you'll see the flagellum waving about instead of it being used to attach itself to the vessel wall. So instead of the flagellum at the front, it actually uses its rear end to attach itself. So at this point you're probably thinking, great, so those squiggles on my screen were upside down the whole time. How exactly does this impact my life? Now, first of all, obviously, let's hope it never will and that you won't get infected, but if you did, you'd probably agree that it'd be nice if we had some sort of cure. A cure against a pathogen like this is often designed to interfere with some crucial system, a unique target, and in this case that could mean targeting the proteins used for movement or extravasation, leaving the bloodstream. A little something we already know from an earlier study is that there's a protein right here that appears to be concentrated in the same area that is used for attachment and might be a good candidate for further research on treatments. A second important advantage is that this new method allows for new ways of testing treatments. We've already seen that the parasites are able to leave the bloodstream, and if you would only be able to look at the blood, you would not be able to say with certainty that a treatment would have been 100% effective. This new model, while not genetically identical to humans, could still provide a more accurate representation than only using blood tests. So because of all that, we now have new insights in trypanosome movement and behavior, but really the take-home message here is that these are findings we never could have had if we hadn't seen them inside a live host. And I'm not saying using artificial models is a useless endeavor. It's important to use artificial means whenever possible, But we do have to recognise that there are limits, and especially when potential medical uses are involved, I feel that it's really important to, at the very least, test your findings in a live model. After all, as shown in this paper, seeing is believing. Thank you for watching CBI Online, and I hope to see you next time.